Hello students, in today's class we are going to study about mitosis. So the aim of the experiment is to study the mitotic stages using permanent slides. Requirement for the experiment includes permanent slides with stages of mitosis and a compound microscope. Procedure includes placing the slide on the stage of a compound microscope and first observing the slide under low magnification so as to find the dividing cells. Once we find the dividing cells, then we change the magnification of the objective lens to 40x and then we observe it under 40x magnification and identify different stages of mitosis. Coming to the observation, the slide shows different stages of mitosis which are explained as follows. Interphase, this is the phase or the stage that is prior to the actual mitotic division. The cell here appears to be inactive or in a resting stage, but actually it is metabolically in the most active stage. During this stage, the DNA is replicated. The nuclear membrane and the nucleolus are distinct during the stage, and the chromosomes are in a form of a chromatin network, and individual chromosomes cannot be seen separated during this stage. Each chromosome is made up of two chromatids. So prophase represents the first stage of the mitotic division. When you observe it under the microscope, you will find that the nuclear membrane appears distinct as well as the nucleus is clearly visible during this stage. The chromosomes, they begin to coil and condense and become shortened and more distinct during early prophase. During late prophase, the nuclear membrane and the nucleolus have partially or completely dissolved or disappeared. Each chromosome by the end of the prophase or during the late prophase appears as more dense and condensed. Each chromosome now begins to show two chromatids, a primary constriction, a secondary constriction, and centromeres are also present. The equatorial region begins to be clearly visible in the center of the cell. The chromosome begin to move or gather near the equatorial plate. The chromosomes are condensed, short and thick, and the spindle fiber also appears to begin or start forming during late prophase. Metaphase is the second stage of the mitotic cell division. During this stage, the nuclear membrane and the nucleolus have completely disappeared, and the centromere of the chromosomes are arranged on the equatorial plate, and each is attached to the spindle fiber. Centrioles are absent in case of plant cell, and hence aster formation is not observed. And this type of mitosis in plant cell is known as anastral mitosis. The spindle fiber is made up of only fibers, indicating that they are plant cells. The chromosomes of the metaphase appears very distinct, and the number and morphology of the chromosome is studied at this stage. Each chromosome shows presence of two chromatids, centromere, primary constriction, euchromatin and heterochromatic regions, etc. can be observed. Anaphase represents the third stage of the mitotic phase, and it is lasting for a very small period of time. The centromere of each chromosome splits into two, and each chromosome also gets divided into two chromatids. Each chromatid now bears a centromere. The chromosome appears short and thicker, and the separated chromatids are now pulled towards the opposite poles due to contraction of the spindle fibers. During the moment, each chromosome shows a characteristic shape such as V shape or L shape depending upon the position of the centromere. Telophase represents the last phase of the mitotic division. The chromosomes are present at both poles of the parent cell. The chromosomes begin to increase in length and become thread-like. All the chromosomes together form a chromatin network, and they lose their individuality here, so that we are not able to distinguish the individual chromosomes here. The group of chromatic network at each pole is surrounded by a nuclear membrane, and a nucleolus also begins to appear. Thus, at the end of the telophase, we are able to see two fully formed nuclei, one at each pole of the parent cell. Spindle fibers are completely absent, and we can also see formation of a cell plate which will follow division of the plant cell. Coming to the precautions, while handling of the microscopes and performing the experiment, we must make sure not to touch the glass part of the lens with the hands. Lens paper must be used to clean the lens if required. We make use of the stage clips to hold the slide in place. While holding the microscope, we need to make use of two hands, one hand at the base of the microscope and other at the handle of the microscope. And handling of the microscope should be done with care. So that's it about mitosis. Thank you very much, and kindly like, share and subscribe if you like the content.